I am Jasmine Masters and I have something to say. I I am depressed. Clinical depression. I am seeing a therapist. I am taking pills. I am diagnosed. What is depression? What does it feel like? Usually people would say it's emotional and mental pain. But then I have also started to experience these physical aches everywhere. They say that it is a symptom of depression, but I've been taking these pills and none of them are working, and I think there might be more to it than just my mental health in general.
Make sure to take your pills and get adequate sleep.
Josie,
Yes.
I'd like to ask for your favor. Could you please fetch these books from the library, please?
Okay. Jasmine, wake up. There's a few books. Yes, yes. Hurry.
Who are you?
I am Lucy.
Where am I?
You're in your house. I guess it's that.
Miss Masters, you have been diagnosed with the Creutzfeldt-Jakob disease as you have manifested 5 out of 5 of its symptoms.
What's that?
Here is the result of your MRI scan. As you can see, you have developed holes in your brain, and this is caused by what you call prions that cause the misfolding of your proteins. And since the disease targets the brain, it's fatal. And I'm sorry to tell this, Miss Masters, but you only have a few months to live.
I am Jasmine Masters and I have something to say. I have all these notes, as you can see, because I now forget. Not forgetting like forgetting what I had for dinner or running an errand that I was supposed to do, but forgetting a specific person, a specific place, a specific event, an entire fraction of my life. And I am recording this now because I fear that it will get worse. So I need to be able to get it out here before it completely leaves me. So I went back to my doctor today, and it turns out that after all this time and all those misdiagnoses, I have VCJD, short for variant Creutzfeldt-Jakob disease. It is a disease so rare and hard to diagnose, and it has no cure. It targets the brain, and so as a researcher, someone who suffers from it, I would love to be able to contribute in finding a cure for it and helping the others. So I did a study on it, and here it is.
A study by Wang et al. In 2011 states that humans and bovine prion proteins are closely related based on structural analysis. However, according to another study by Reed et al. In 2003, if the genetic sequences of humans and bovine prion proteins are evaluated, the human prion protein will have more similarity with primates because humans are more closely related to primates based on ancestry. and the cows are more closely related to sheep and other bovines. This does not explain how the prion diseases are transmissible from cows to humans. Using bioinformatics, a field of science based on computational tools and molecular data, I evaluated the genetic data of the alternative prion protein of humans. Since based on the study of Van der Peer, these proteins are co-expressed with those of the bovines. The molecular data of the alternative prion protein will be formatted in a FASTA format, copied to stickies, and will be subjected to the BLAST program. The program will show the organisms that have genetic data that is closely related to the alternative prion protein of humans. The genetic data of all of the related organisms will be formatted in the FASTA format and will be copied to stickies. Each of the genetic sequences will be renamed based on the scientific name of each of the organisms. The MEGA program will be used to create a phylogenetic tree of the organisms. By opening data, the stickies file can be converted into a FASTA file. By clicking the Align button and edit/build alignment, the sequences can be aligned by clusterdobly. Then, the sequences of the other organisms that is not within the range of the reference sequence or the Homo sapiens will be removed. The file will then be saved into a FASTA format. The Models button will then be selected to find the best DNA or protein models. The resulting window will display the VIC values of the data, where the lowest VIC value is the best model. To create the phylogenetic tree, the phylogeny button and construct/test maximum likelihood tree will be selected. The FASTA file will be designated, and the test of phylogeny will be the bootstrap method. And the rates among sites will be based on the results from the models. Now, a phylogenetic tree based on human alternative prion protein was constructed.